Good morning, I'm Matt Galloway, and you're listening to The Current. We're off to the laboratory.
Hi.
Hi.
Jennifer, right?
Yes.
All right. Shot from the past, right, Jennifer? All right.
The camera's clicking as Jennifer Haller becomes a human guinea pig. She is the first person to be injected with a possible COVID-19 vaccine, and all of that happened earlier this week in Seattle.
There's the usual vaccine kind of potential side effects, soreness at the site of injection, fever, chills, nausea, things like that. And I actually have not experienced those at all so far. In fact, my arm feels better than it has with a previous flu shot. So, of course, this has never been tested on a human before, but I'm doing great. Everything's good.
Everything's good. Here in Canada, there are also teams working towards a vaccine, teams that include Alyson Kelvin and her colleagues. Alyson is an assistant professor at Dalhousie University and a researcher with the Canadian Center For Vaccinology. Alyson, good morning.
Hi, how are you?
I'm well, thanks. What do you make of this, what happened earlier this week in Seattle, where, as I mentioned, Jennifer Haller essentially becomes a human guinea pig?
Well, I guess it's interesting, and of course, there's a lot of compelling reasons for putting out a vaccine right away. As we've seen the SARS-CoV-2 virus spread throughout the world, there's a lot of unknowns so far about this vaccine, and all I can do is wish it the best because we really need a vaccine right now.
What stage is your team at in developing a vaccine for COVID-19?
So Dalhousie has what we've called a COVID-19 Rapid Response Team. And this is made up of researchers at Dalhousie University and the Canadian Center For Vaccinology. We have 4 different vaccine platforms, and this means 4 different vaccine types. So the first one was developed by Roy Duncan, and he has what is known as a DNA vaccine. So that enables your own body to make the viral protein and produce antibodies to it. And the good part about DNA vaccines is that because it's just DNA and it uses your own cells to make the protein, we can scale it up really quickly.
So you mentioned there, and I guess that's the first of 4 of different kinds of vaccines?
That's right.
Yeah, okay.
That's right. So this particular one, we're looking at possibly having vaccines ready to test in the next 3 days, which can make it to human clinical trials in the next 3 weeks.
Oh.
The other vaccine platforms were developed by another researcher at Dalhousie named Chris Richardson. So his vaccines are more protein-based. So again, these are based off of pieces of the virus, but they're proteins, so they're not just the genetic code, and which we will inject into a person and hopefully induce an immune response.
How long is this process? If you're talking about where you're at right now, what is the timeline for this process?
Right. So typically a vaccine development will take over a year. And that's because, first you have to start with the genetic code of the piece of the virus that you think is going to induce an immune response in a person. So again, we want to make those neutralizing antibodies to block infection.
Okay.
So after that, either if you go with a DNA vaccine, this can again be done quite quickly because all we're doing is making the genetic code and then using a person's own cells to make the protein. So your own cells become like factories making this protein that will hopefully induce an immune response. Whereas for a protein vaccine, we need to make the protein, and this takes a lot longer to kind of scale up the protein in the lab. And then, of course, we like to test the vaccine in animals. I use ferrets because for this particular virus, the SARS-CoV-2 virus, ferrets, we hypothesize, will be susceptible to this virus. So they'll be infectable, which means we can immunize the ferrets and then see if they're protected from this virus challenge later.
It sounds like with these different types of approaches that there's a lot of collaboration with other researchers. How common is that?
Well, it's common, but you know what? Right now, in this state of emergency that we're in, I think all the scientists are ready to collaborate and move this forward as fast as possible. So we really see the urgency, and in that way, it's been a really good time for science and kind of that collaborative atmosphere.
But in the midst of that collaboration, you're also working in the middle of a pandemic. How does that affect your work, given what we're all involved in right now?
You know, it's been trying times, even to go out to the grocery store, which means that it's harder for us to get in reagents that we might need to make these vaccines and test them. So even getting in our DNA or kits that we might need to assess the immune response, it's harder because, to even begin with, some of these reagents are typically made in China, and we know China is going through a hard time right now, so it's harder to get those reagents from China here to Canada. And now with a lot of the shutdowns, it's harder for movement inside North America.
What about in the lab itself? I mean, are labs staying open? And we keep being told that you need to social distance. Can you social distance within a lab as you're trying to work on a vaccine?
Well, we're doing our best. A lot of the labs have started to ramp down here at Dalhousie, but there's been a prioritization for COVID-19 work. So we're trying to manage using social distancing practices while being in the lab. So this is cleaning surfaces often and maintaining a certain distance from people, not coming in, obviously, when you're not feeling well, reporting symptoms. So doing what we can to make sure that we're getting this vital work done, and but also keeping the community safe.
What sort of pressure do you feel in the midst of this work? We keep hearing world leaders, health experts, the average person who's worried about this talking about a vaccine and talking about the need for a vaccine. You're in the midst of this work. What kind of pressure do you feel?
Well, I feel pressure, but really I feel an overwhelming sense to want to help. So I guess that's the biggest feeling I have. And it's hard to see your neighbors and your colleagues, be sick. I have lots of colleagues in China and in Italy, and it's horrible to watch what they're going through right now. So I wish I could do more.
But in the midst of that, I mean, maybe it's not pressure then. You feel like you have an opportunity or a responsibility to help, for example, those colleagues in China and in Italy? That's right.
I really do feel like this is what my job is for. I think about pandemics a lot. All of my work revolves around what could be the next pandemic. So even though that was skewed to more towards the influenza virus, all my work is easily applicable to this current situation.
Just before I let you go, this is a difficult time and a dark time for a lot of people. What is giving you optimism in your work?
Again, that sense of collaboration and wanting to push the science forward. There's some good candidates for antivirals, but also the community. So I look out and see a lot of members of the community trying to help, either by practicing social distancing or making social distancing practices in their own, businesses. So you see a lot of that, and I really hope everyone the best.
These are long days, I'm sure, for you. I appreciate some of your time today. Thank you.
Thanks for having me, and I wish everyone well.
Alyson Kelvin is an assistant professor at Dalhousie University and a researcher at the Canadian Center For Vaccinology. She was in Halifax.